Going to have a class on our snow day, and I grew up in Minnesota, so I had a lot of snow days as a kid, and I know how special they are. And I'm not having a snow day today. I'm also happy to talk about science. So here we go. So today, Thursday the 6th, maybe, we were going to have Sam talk about the first step in CAR T cell therapy, and we'll have Sam do that next Tuesday at the start of class. And we'll talk at that time about what the setup is there. I talked on Tuesday about these summer internships, paid internships, fantastic experience. And so check it out. You can talk to the organizer, Samantha White. Sam works in the building, and just make a connection, and Sam will explain to you everything about the opportunity. This is what you should do this week, and we have an addition. So here in red is the addition. There's a homework assignment, and so I'll post the link for that. It's to watch the video that I call Wisconsin to Boston, even though that's not irrelevant at all, and you don't have to remember that detail. It's about a drawing assignment in which you're going to make 3 drawings that are in the video. And so simple labeled drawings, and by a week from today, before class time, because we want to talk about these topics in class time on next Thursday, you need to send me pics of your 3 labeled drawings. Yep, and you save your drawings. You don't turn them in because you're going to be able to study from them before the exams and stuff. Okay, so Wisconsin to Boston tells a story of why our textbook author first moved to Wisconsin when he was your age. Discoveries were made there, and then he moved to Boston, and he shifted his experiments. In Wisconsin, he studied a retrovirus, and he stopped studying it when he moved to Boston. What he continued to study was a gene called RAS. And so watch the video and complete 3 labeled drawings described in the video. Take 3 pics and send them to me in an email before class by next Thursday, the 13th. Okay? All right. So we talked last week a lot about how discoveries get turned into new tools. One of the drawings in the Wisconsin to Boston assignment is another example of how a discovery got turned into a tool. So that's a real common story in molecular biology. And the reason we talk about those stories is because they help illustrate and introduce the concepts. So remember, we want to emphasize, these clay model videos, I made those out of clay because scientists can cut those DNA pieces up in a dish, in a test tube, and rearrange them any way they want. And so the ability to do that led to utilizing discoveries like GFP, green fluorescent protein, and how that can be used to create reporter transgenes and transgenic animals, and lots more. Great. Okay, so just an image of that to remind you of that. Again, prerequisite videos that we started our course with, from that jellyfish and into transgenic organisms like this zebrafish. Okay? So that's that. And then another drawing that we did last week, and we did several drawings, was to introduce the idea of gene expression delay based on the regulation of a transcription factor. And that's a central concept in our course, is that we inherited from our parents just a fixed, really relatively unchanging DNA sequence, and but it's dynamic, right? All through our embryonic development, fetal development, childhood development, puberty, adulthood, the gene expression is changing. It's changing over time, then it's changing in location, in the insulin being made in the pancreas, hemoglobin being made in the precursors to red blood cells. Yeah, it's just a central story for our bodies, and it's a central story for every cell on Earth. Every cell on our planet has DNA as the primary heritable molecule, and every cell inherits more DNA than it uses at one time. So the concept of switching gene expression on and off is one of the oldest concepts and a shared concept of every cell of every living thing on our planet. Yep. All right. So now we're going to just talk a little bit about Chapter 1, and we're going to kind of wrap it up. Okay, so here's a little bit more on that, and that is in our Chapter 1, it talks about model organisms. And so here's a set of model organisms, and here's the key concept for us, for model organisms. When you say, why are these organisms studied? A person will say, well, fruit flies grow fast and reproduce quickly. And that's a feature of why that species was picked on, especially. And all these organisms have special features that make them amenable to studying in a research lab. But here's the key thing. They all have orthologs. That's why they're model organisms. If a fruit fly wasn't closely related to us, using orthologs that we could easily identify, no one would study fruit flies. Who cares about fruit flies, right? Yeah, it's all about orthologs. So when somebody says, hey, why do people like to study fruit flies or mice? Then you just say it's all about orthologs. It's all about that concept. And remember, some biologists are a little bit uncomfortable talking about it because we can never prove that last common ancestor had that gene. We can't study that gene because it was hundreds of millions of years ago. But remember, the structure and function are the same in the two species. And then when we compare their protein sequence by BLAST, they're so similar, we can't come up with an alternative explanation. So here is another example of a model organism that's emerging, and that's just organs in mammals. So a kidney as an organ is now becoming a model organism. And because of the tools that can manipulate the organism and that organ are becoming so powerful. So this is GFP, just tagging the different developing nephron units in a kidney and helping scientists understand kidney structure, function, and development. Okay. Another idea of a model organism is that some organisms that can't really study, for example, humans, provide opportunities to raise just billions of cells in dishes like this under sterile conditions with liquid nutrient media and study those in a dish. And so that's another really common controlled experiment that are done, and that brings humans in as a model organism opportunity. The final thing we want to talk about is there are lots of viruses, and molecular biologists initially created molecular biology as a field of study back in the 1940s by focusing on specific viruses that infect bacteria called bacteriophage. And then they studied the bacteria. And so molecular biology is a very reductionistic science, and when we want to sum up the whole that the parts are made of, we want to talk about simple systems. And so just to show you this here, we're going to have some viruses in that Wisconsin to Boston homework assignment that are called oncogenic retroviruses, and they're a special mutation of normal retroviruses. HIV is a retrovirus, but it's not an oncogenic retrovirus. People who have AIDS can get cancer, but not directly. These oncogenic retroviruses would cause direct, immediate cancer formation of the cells they infect. And so we'll talk a little bit about some of these other viruses. Here's the one that I mentioned already. A bacteriophage is one that infects bacteria. Here's a picture of bacteriophage infecting bacteria. You can see it has this black protein coat, and then it has its genome, and its genome could be made of DNA or RNA. Bacteriophages' genomes are made of DNA. In the 1940s, they showed it was the DNA that went into the cell to infect it. So DNA is the heritable molecule for a virus, and those early molecular biologists discovered that, and they were the reasons why Watson and Crick went after the structure of DNA with such enthusiasm. They were studying the bacteriophage infecting a bacteria, and the species that is the model organism is E. coli. And everybody's heard of E. coli because there's a strain of E. coli that can make people sick. Normal strains of E. coli are in our body all the time, in our digestive system, and all the time. Lots and lots of E. coli in our digestive tract. And scientists just picked that as a model organism. And I don't know if anybody even remembers the history or can trace back the history of why E. coli became such a popular one, but it's really E. coli. And this is just called a streak plate. The scientists took some E. coli and they just spread it out on this plate with a little microbacterial loop, and then grew the plates in incubator overnight. Here's just an interesting picture to see them studying bacteriophage. So a bacteriophage can infect E. coli and they can kill it. and destroy it, lyse it, break it open. And so here's a plate, and this plate has E. coli in it, but it wasn't spread as a streak plate like I just showed you with individual colonies. What happened here is that the scientists mixed lots and lots of E. coli with some melted agar, poured that on a plate, and grew that. And as a result, this is just a complete coating of E. coli. It's called a lawn. And what you can see here is a series of plates that are titering a bacteriophage. And they have increasing amounts of bacteriophage because the bacteriophage make these little circles where there's no bacterial growth, and those are called plaques. And it just shows where one bacteriophage went through its replication cycle of infection, and killed all the bacteria in that area while the lawn was growing. So you can see this increasing number of plaques over the plates. And so this is just an example of how the scientists would study bacteria on the plate, or the phage, the bacteriophage that infects those bacteria on a plate. Yep, so again, back to that. And the infection cycle is really fast. So as the bacteria is trying to grow and form that smooth lawn of billions of bacteria, the bacteriophage in that location kills them. And it's just an isolated plaque because neither the bacteria or the bacteriophage can diffuse very far in that agar that is the gel that they are growing in. So last thing I want to just overview is I'm not really going to talk about the tools of cell biology very much. We already talked about centrifugation. We talked a little bit about an electron microscope, but you're expected to read that and learn from it. And so I'll just say, there's pictures of light microscopes. There's description of features of light microscopes. There's pictures of electron microscopes. There are pictures of using fluorescent microscopes. This is a picture of using something that's called DIC, differential interference contrast microscopy. You should, and it's a type of light microscope. Then this again shows a DIC light microscope image versus a fluorescence microscope image. And you should just read through the chapter as you already should have done, and just learn as much of that as you can, because there can be questions on the test about that, and you're welcome to ask questions in class to clarify what's described or ask what we are expected to learn about. Here's information about finding the different organelles in a cell by fractionation and centrifugation. We talked about that. Our favorite example is to isolate nuclei and use a Down's homogenizer and then centrifuge. There are some kind of fairly complicated methods that are described in Chapter 1. FRET is an example. And I could ask a question, 2-point question. What is FRET? How does FRET work? And what would it be used for? And you're welcome to ask questions in class, or we can meet and you can have questions written down and clarify them. That would be great. Yep. All right. So now we're going to start going into Chapter 7. And we're going to organize Chapter 7 in a way to drive through it, getting a really solid, big core of the knowledge from Chapter 7. But rather than just show one PowerPoint image after another, I'm going to describe this in a different way. And I'll use examples that we've already discussed and introduce a couple of new examples. So there are 3 major ways that transcription factors regulate the rate of transcription. So remember on our quiz, I think our second quiz, what do transcription factors do? And the answer is regulate transcription. What is transcription? Transcription is the transcription, the copying of the transcription unit by RNA polymerase. So this is about how do they do that? And there are many steps in the process. So the first thing is we have to describe something called a rate limiting step. And when I ask classes that about if they know what that is, they do, because there's a year of intro chem as a prerequisite for our course. And in intro chem, you learn about multi-step pathways and the concept, that there will be one rate limiting step. Okay? And so the example I always think about is I like to think about when you go to like a county fair and there are booths that you can walk up to, and the booths are just open and they are selling food like cheeseburgers at these booths, and the booths are run entirely by volunteers. It could be a church group, it could be like a high school sports team, or a community group of some kind. And so it's just folks that are friendly, a bunch of friendly folks, and you're in line, and the line can be long. And so as you're waiting in line, you can see into the booth and you can watch them making burgers. And making burgers is a multi-step process, right? So starts out, somebody is cooking the burgers, then there's somebody putting cheese on it. And that's usually how they set this up. They have plenty of people volunteering, so they try to find a job for everyone. So somebody's setting that up. Over at another table, somebody is slicing tomatoes and bringing plates of tomatoes over to add to the top of the cheeseburgers as they finish. There's a pickle lady who adds pickles. Somebody adds lettuce. Somebody adds toppings like mayonnaise or mustard and so forth. And so it's a multi-step process, and then they bring it up to the counter and they hand it to you, and you walk away. Okay? So that's what transcription regulation is like. Transcription is a multi-step process, and for a specific gene, and we're going to start with myogenin. What is the rate-limiting step in myogenin? Because the rate-limiting step is the slowest step, and that is the step that the transcription factor is going to affect. Here's the example back to the booth. If there was a really long line, and you were watching the people making the burgers, and you saw the pickle lady, and pickle lady was very chatty and just doing her job slowly, and before her, there were just a whole bunch of cheeseburgers lined up on the table. So the cheeseburger fryers and cheese appliers are making them faster than the pickle lady. And then after the pickle lady, there are also no lines waiting for tomatoes and so forth. The pickle lady is the rate-limiting step. And if you want to complain, right, when you get up there, and people do, and you just hear people come up there and they say, why are you guys so slow with these burgers? I've been waiting 20 minutes. And you complain, you need to make them faster. That's not helpful. But if you target the rate-limiting step and you say the pickle lady needs help, that could increase the rate. If you say you guys need to get more cheese, that won't help at all. Imagine that the line in front of the pickle lady of cooked cheeseburgers just gets bigger. Imagine you say you need a second table to have those cooked cheeseburgers waiting for pickles. There could be 2 tables, and that won't help the burgers get served fast. Yep. Okay. So going through that analogy, this is what's going to happen. So the myogenin step is early in the process. So myogenin is going to be affecting something like helping unwrap the burger patties. The estrogen receptor is going to be something like opening the booth up. The booth is closed down, and we have people waiting in line, but we haven't even opened the booth yet. Then this heat shock transcription factor, which is a new transcription factor that we'll add to our list because it's a very interesting protein, and there are orthologs found in humans and every other eukaryote, all animals, all plants, and even single-celled eukaryotes like the yeast that causes bread to rise. So heat shock transcription factor is in a lot of different cells on our planet, not bacteria and other prokaryotes, but everywhere else like that, okay? So this one is very late, astonishingly late. The rate-limiting step is the person who's handing you the cooked burger, and they're chatty, and they won't let go of the plate. So we have different rate-limiting steps. Why? Because these are different target genes. Each of these is a different set of target genes, and they just have a different rate-limiting step. So that's what this is about. So on to our first one. So we don't have to review that at all. We are experts on MyoD and myogenin. And here's what's happening with that. We can go to our original clay model drawing, and if we do, there's the clay, and we have the transcription unit, we have the promoter, and we have the enhancer. And the enhancer is going to have MyoD bind, and MyoD is going to bind as soon as it gets made, and it's going to activate myogenin transcription. And how is it going to do that? It's going to increase the rate limiting step. And what's the rate limiting step? The rate limiting step is something that needs to happen on every gene, and that is on this promoter. We won't talk very much about the transcription factors on this promoter, but we're going to name one of them here in this example. There are you could say there are a dozen different transcription factors that bind here, and these are binding to the promoter. So this is DNA, and it's the promoter. And we know that these are parts of the cis regulatory region. And I've said that people have some different names for these regions of the regulation. And another name for this promoter is, it's sometimes called the proximal promoter. And that's because it's close to, and in fact it's right at the start of transcription, and people refer to the start of transcription as +1, because then they just count the nucleotides in the transcription unit like that. The RNA polymerase is always going to go this direction. Then this, the enhancer, and we've seen that people don't like that name when they're really working in the field. That's fine. This can also be called the distal promoter, and that's a nice term. Proximal and distal. Another name for this promoter that's used more often, because it talks with kind of emphasis about the function of these, is this is often called the basal promoter. And I know these are a lot of terms for the same thing, but that's okay, because this is what we're really into in our course. So the basal promoter is just, basal means minimal, and that's another thing that sometimes people say. And it also means kind of the basic reporter. This doesn't typically regulate the transcription. This typically isn't involved in the big stories of gene expression, but it has to get assembled, and it's going to get assembled here along with the RNA polymerase. So here's the enzyme that's going to do the actual transcription, and we got to get that there. We got to get it positioned, and this promoter, and I'm usually just going to call it the promoter. That's fine. That has to assemble these proteins, and all together, these set of proteins and the RNA polymerase are referred to as the pre-initiation complex. And that's a nice name. Initiation means the start of transcription. So you can think of it as the RNA polymerase landing. It's getting ready to do its job, and now it starts doing its job. And in the myogenin gene, there is just one protein that's missing, and it's this one. And so we're going to zoom in on that. So in the pre-initiation complex, all the proteins are assembled except for that one. And I'll just redraw this, and we'll zoom in on the promoter region. So this is +1, and that's the transcription unit. And there's just one protein missing, and it's going to be this protein that's missing. And the function of the, well, let me just do this out here, because there's the enhancer, and there's MyoD. And MyoD protein is going to, this DNA is going to get bent, so that the MyoD protein is actually going to bend and get close to that region. And the MyoD protein is actually going to help bring this missing protein. And the missing protein is part of the pre-initiation complex, and it's part of the promoter binding transcription factors. And this protein binds to a sequence in the promoter DNA. So that's DNA, and that's the promoter, the proximal, basal, minimal promoter, and we need to get that protein there. And the sequence in that region looks like that. So people have referred to this because it's a little bit longer, but it basically looks like that. And people call that the Tata box. And many, certainly most promoters have a Tata box. And luckily for us, the protein that binds there has a great name. It actually has multiple names because multiple scientists studied it, but it's called Tata binding protein. And on promoters, which are the majority of promoters, they need that Tata protein to get there. And a lot of promoters, it gets there by itself really quickly. It's not the rate limiting step. But for myogenin, it's the rate limiting step. And so that's what MyoD has to do. How does MyoD activate transcription? It helps get the Tata binding protein to bind to the Tata box and complete the pre-initiation complex. So you have to be able to explain all that on a test. That's how MyoD works, and we love MyoD. And that's the rate limiting step of the myogenin gene. What's the rate limiting step of the myogenin gene? Okay, so then these two proteins, they bind together. They interact, they bind together. The MyoD protein grabs the Tata binding protein like that. And it helps it because it's tethering it, right? It's like tethering it to this area, and it will help deliver it to this. And so that's what's going on here. It helps complete the pre-initiation complex. And then RNA polymerase goes down the transcription unit, and we have to assemble the pre-initiation complex again. So over and over again, MyoD is helping get the Tata binding protein, and Tata binding protein is abbreviated TBP. And again, there are a couple of other names for it, but that's a really nice name for you to remember. Okay, so that's what's going on, and that's our first story. The rate limiting step in the transcription of myogenin is TBP is missing from the pre-initiation complex. The other promoter binding proteins and even RNA polymerase are there ready to go, and we just need to get the TBP there, and MyoD's job is to do that. It's the rate limiting step, so MyoD must act on the rate limiting step to increase transcription. Transcription is off. We need to increase the rate of transcription. No burgers are getting cooked, and what is the problem? Well, the pickle lady hasn't even gotten here yet. And you're like, can't anybody else do the TBP job? No. We have to wait for TBP to get everyone on the same team and go. And that happens over and over again every time you have to get RNA polymerase to the transcription unit. So here is the Tata box, and here it's showing a complex of proteins binding to an enhancer, and it's showing how, yes. And so this is the pre-initiation complex that's assembled. This protein right here is actually TBP. I mentioned that it has another really famous name, transcription factor 2D, but we don't have to learn that name ever. TBP is great. And you can see there are lots of proteins at play here, and this is the one that is the rate limiting step. This is cheese, this is the burger, this is the bun, that's tomatoes, that's ketchup, and so forth. And these are the pickles. This is the pickle protein, okay? So Tata binding protein is a general transcription factor that binds specifically to a DNA sequence called the Tata box. This DNA sequence is found about 30 base pairs upstream of the transcription site in some eukaryotic gene promoters. Right? Is that what we're learning in our course? Wikipedia writes for college student learning. Yep. And then it goes on to say, well, it's actually a gene family. And so sometimes the promoter story and the pre-initiation complex story becomes kind of complicated. In our course, we're going to take it as being pretty boring and pretty common. And instead, we're going to really focus on proteins like MyoD that bind to enhancers, because those are the proteins that really typically regulate the rate of transcription. The reason we're talking about the promoter binding proteins right now is because sometimes they're not all assembled. It could be TBP or something else. We're talking about TBP because we love MyoD and we understand MyoD well. Okay, now just look at this. TBP, dependent activation of MyoD target genes in skeletal muscle cells. That looks like a title that helped make the discoveries for what the clay model videos taught us. Then look at this, because it says that TBP also helps MyoD bind to its site. So they help each other, and that's a little bit of a surprise. So what we're saying here is sort of like this, is that before, so here's some DNA, it's going to be folded like that, and there's the enhancer for MyoD, and there's the promoter with the Tata box. And what that scientific article seemed to show is that what actually happens is MyoD binds to TBP before either bind to the DNA. And what happens is that together they find their binding sites and they help each other. And so that's just a fascinating detail, and I just want to share that because it's fascinating to see these molecular relationships and the details of these molecular relationships. And it's kind of impossible to share with an advanced undergraduate course, right? This is a college course for seniors, to share just how much detail is known. We could spend an entire semester on a course that's just called MyoD, because it's just been studied, and so many of these proteins have been studied in tremendous detail. So isn't that fascinating to see that? TBP stabilizes the binding of MyoD to its DNA site. And they're using the promoter in a broad sense, right? The proximal promoter, distal promoter, like there's some people like to talk about it that way. So they're meaning to its DNA site, the enhancer. And in turn, MyoD facilitates the association of TBP with the pre-initiation complex. Again, a slightly different name for Tata binding protein because different scientists had studied it all at the same time and wanted to, sort of promote their special name. TBP is the long-standing name. MyoD targets TAF3, TRF3 to activate myogenic transcription. Same thing, just another name for that's transcription activating factor 3. And these are biochemists, and biochemists do that. They just provide lots and lots of different names, and they just shrug, to figure it out like that. But ours, what do we call it? TBP is great. So TBP. And I think that almost everyone calls it TBP nowadays. These are all papers from 20 years ago like that, approximately. Okay? So here comes the next example, and maybe I don't have it on here, so let me just scroll back and grab it. The second example is estrogen receptor and other nuclear, oh, did I now overshoot it? Yeah, here we go. Okay, so our second example is the estrogen receptor. For the estrogen receptor, what's its rate limiting step? And the story is going to be the booth isn't even open. Is the pre-initiation complex assembled? Is RNA polymerase there? Nope. None of the workers are even there yet. And what estrogen receptors is going to do, it's going to open up the shop, raise the curtain, and get everything, get the fryer going. Raise it up, open it up. All right, and that's a very early step in the steps required for transcriptioning. It's really the first step. And what we're going to learn is, we usually draw the DNA like this. The DNA can be folded up and packed away. Like how you fold up a blanket, winter blankets get folded up and put away in the summer in a closet. So that's what's going on. So the estrogen receptor and other nuclear receptors, and we learned what those are, 48 members, 48 paralogs of a gene family. Nuclear receptors work by first, in the absence of hormone, in the absence of estrogen, they actually cause the target gene's transcription region to be folded up. And then when estrogen arrives, they actually do the opposite action. And they do that because in the absence of estrogen, they recruit an enzyme whose activity is to modify proteins and cause the DNA to fold up and be inaccessible to tau-binding protein and RNA polymerase and any of those other proteins. And then when estrogen arrives, the estrogen receptor, ER, lets go of this enzyme and it binds now a different enzyme that does the exact opposite chemical reaction activity. There are proteins along here that we'll learn about called histones, and those are the target proteins for this enzyme. And in the absence of the estrogen, the enzyme removes an acetyl group from histones, and they just naturally fold up tighter and pack the gene away. And then, so it's a deacetylase, and we'll write this all out. And in the presence of estrogen, this deacetylase is let go, and this acetylase is added, and it's just the exact opposite reaction, and it causes the DNA to unfold. So that's the rate-limiting step, the three major ways. There are a bunch of transcription factors, not just the nuclear receptors, not just estrogen and the nuclear receptors that work this way. So let's take a look at that. So the first thing is we want to make some definitions of terms. And so chromatin is a word for the DNA plus proteins bound to the DNA. And chromosomes are made of chromatin. So we have DNA and we have chromosomes in our body, and the chromosomes are made up of DNA plus the proteins that bound to the DNA. Those proteins include all the transcription factors we've been talking about, and they also include some sort of structural proteins, and the most famous of those are histones. There's lots and lots of proteins involved in binding to DNA because DNA is really long. If we just stretch DNA out into its typical double-stranded helical structure, it would be, in one of our cells, would have about 6 feet of DNA. And that has to get packed into the nucleus. So, so we're going to emphasize, and I failed to switch. The chapter we're reading here is chapter 7. This is from the previous edition. So we've already talked about how we really care about the estrogen receptor because how does the receptor get activated? It's a hormone. And so a ligand is a more general word than a hormone. A ligand just means a small regulatory molecule. And so we could call it a hormone or more generally a small regulatory molecule, ligand. And it binds non-covalently. In the case of the estrogen receptor, the estrogen hormone is a steroid hormone, so it's just a small molecule that binds and regulates the activity of that protein. And this is a great example of regulating the big picture of our story, which is how is it that the genes that we inherit from our parents that are pretty static and unchanging, how is it that they are expressed in a dynamic, changing way? And the answer is, well, transcription factors. Okay, so here's a picture looking ahead, and this is chapter 16, and I got to update my figures to the 9th edition. But this is the next chapter we're going to read. I typically call it chapter 17, and I just haven't, this is the first year we're teaching with the 9th edition, and I just have to update my words. In this chapter, this will be the next chapter we read. We're going to read 1, chapter 7, chapter 16, 17, 18, 19. Okay, and so here is the estrogen receptor, and or in this case, it's another nuclear receptor family member, the glucocorticoid receptor. The picture would be the same if it was the estrogen receptor or the testosterone receptor, and so it's going to bind. And what it's showing here is it's bound with this enzyme that's going to modify the DNA. And because the hormone is present, this is the activating enzyme, and it's called histone acetylase. And that's what we're going to call it. it's just abbreviated here to get it on the figure, HAT, but everyone calls it histone acetylase. And these are little acetyl groups, chemical groups that are acetyl groups, and this enzyme covalently bonds these acetyl groups to the histone. So stable modification of these little histones, and that's going to cause the DNA to open up, and it's going to increase transcription. So in the presence of the hormone, and this would be the estrogen hormone for our example of the estrogen receptor, we're going to activate transcription of these target genes. And we have, and let me just find this. We're going to name one target gene. I bet I don't have it until later, but let's just say it since I mentioned it now. Find it. You see, we want to talk about it a bunch. Oh my gosh. Isn't this amazing how much we have to talk about chromatin and these guys? Maybe it's not even on here. I missed it. Nope, I missed it. So we'll have that by Tuesday, because we're generally going to call these genes, we're generally going to call these genes, have we gotten back here yet? No. We're generally going to call these genes estrogen target genes. And that's generally what we do. Estrogen target genes, myogenin is a target gene of MyoD, and transcription factors often have more than one target gene. We just talk about one as an example, myogenin, but MyoD almost certainly activates a number of different target genes. So we'll have one specific target gene that we'll learn about of the estrogen receptor that gets activated, right, in females, and that's related to, maturation of female reproductive development. And but for now, it's just these are a bunch of estrogen receptor target genes. And so if we just skip over here and draw this out, what we're talking about is instead of this name, we're going to be looking at this being the estrogen receptor response element, or the estrogen response element. That's what the enhancer is. And this is going to be the estrogen protein and the estrogen receptor. And the estrogen receptor has 3 domains that we'll learn about, like this. And the first domain, and we need to learn this, and so nuclear receptors are made of 3 domains in general. And one domain is called the DNA binding domain. And another one of the domains is called the ligand binding domain. And the third domain is called the trans-acting domain. And so this is going to be the domain to which that enzyme binds. So this is going to be that histone acetylase enzyme. And I draw it as acetylase enzyme because it's going to have a chemical reaction that it catalyzes. So that's the histone acetylase. And of course, this would be an example of an estrogen target gene that's going to get activated. And so this, for its substrates, has histones and acetate. And it catalyzes a chemical reaction where the histones, the acetyl groups are added to the histones. And so we have acetylated histones. And the acetyl group just changes how these fold up. And histones are part of the chromatin that binds to DNA and helps it fold up. And this unfolds it. It opens up the burger shop, like that. Opens it up for binding all along here. So now we can assemble the pre-initiation complex because we've unfolded the DNA so proteins can bind to it. So it's a physical, accessible story. And so I'm just going to redraw this protein and I'll show you this. So this is what a nuclear receptor looks like. It has these three domains, and we'll draw them like this. And so the DNA binding domain, the transacting domain, and remember that this receptor is a protein, so there must be somewhere else a estrogen receptor gene. So this gets made, and this protein acts in trans on estrogen target genes. So this is the part that is acting in trans. Remember the terms cis and trans. This is acting in trans. And this is the regulatory region that's going to affect the rate limiting step. And the rate limiting step is, in the absence of receptor, these are all folded up and put away. And then over here is the ligand binding domain. And see this shape? This is just the hormone binding site. So estrogen is not here. So there's estrogen. It's going to fit in right there. It's not here yet. Okay? So in the absence of estrogen, this transacting domain has a certain shape. And there's a protein that binds to it. And in the absence of hormone, this protein is called histone deacetylase. And its substrate is acetylated histone. And its products are histone and acetate. So that's what it's going to catalyze. And it's bound here in the absence. And so the estrogen receptor is here, and it is recruiting and delivering it. Now, the next thing to mention about this is, remember that this ERE, estrogen response element, DNA, that can act at a distance, and it can act at either orientation. It can even be downstream of the gene. Because what's happening here is we're tethering it. And what are we tethering? And the answer is we're tethering this enzymatic activity to this region. So this is flopping around like it's on a leash, and everywhere along the length of the DNA, the histones are getting deacetylated. All along here, and it's thousands and thousands of nucleotides long and probably dozens and hundreds of histones, and this enzyme is targeting them all because this enzyme has a high local molar concentration because it's tethered to the estrogen receptor element DNA, and that DNA is what's tethered to the gene. And we're going to change the chromatin. And so this procedure is an example of many, many types of examples of what's called remodeling chromatin. And we're remodeling it because you see it takes several minutes. So we're rearranging it, and we're opening it up. Chromatin. Oh, in this case, sorry, we're closing it. I may have been misspeaking this whole time. No hormone, so we're folding it up. It's getting all folded up all along here. So no opportunity to bind by the tau binding protein or the other promoter binding proteins or RNA polymerase because it's all folded up all along here. When there's no hormone, this folded up chromatin is called closed chromatin. And isn't that a great name? It's also called heterochromatin, and we need to learn that term as well. And there are two types of heterochromatin, and just to give you the right out of this, so we're looking at the heterochromatin, And the heterochromatin is chromatin. Remember what that chromatin word means, DNA and proteins. Heterochromatin is folded up. And there are two types of heterochromatin. When we look at a chromosome, here near the center, there are no genes because this is just a structural region at the centromere. And this is called constitutive heterochromatin. It's always closed. Constitutive heterochromatin. The estrogen target genes are called facultative heterochromatin, and that means they can change. In the absence of estrogen, it is closed heterochromatin. In the presence of estrogen, it's called open heterochromatin, unfolded, transcriptionally active. So that open chromatin is transcriptionally active. And, not heterochromatin. The unfolded chromatin is called open chromatin or euchromatin. So the two terms are closed heterochromatin and open euchromatin. Yep, and so we just need to learn those terms. And the rate limiting step for estrogen receptor gene expression is that it is closed chromatin. Not constitutive heterochromatin, but closed facultative heterochromatin. It can be unfolded, and that's what the estrogen receptor does. So let's go back to here and walk through some of these slides. So here is the closed silent heterochromatin. Here is the active open euchromatin. Gene repressed, no transcription. This is what's happening in girls and males. And then euchromatin, active chromatin, open chromatin, transcriptionally active chromatin. This is what's happening in adolescent girls. Here's a picture of histones. There are 4 histones, and these 3 make this core of histones that's called nucleosome. And you can see their function is to have the DNA wrapped around them, and that helps pack them. And then there's also this additional linker histone, and it binds to the linker DNA. So this is what a nucleosome is like, and about 200 base pairs of DNA are wrapped up. And this could be wrapped up tight and be heterochromatin, or more open, looser, and it'd be open euchromatin. And the difference is that each of these histones can get modified. There are lots of different ways that histones can get covalently modified. Acetylation is the only one we're going to read about. Chapter 7 lists a bunch. So acetylated histones are our favorite for our course. Acetylated, open, deacetylated, closed. So chromatin remodeling, and it's a modification of the DNA and the proteins bound to it, and it could be condensed, closed up, or open. So there are 3 major ways that transcription factors regulate the rate of transcription, and we're talking about the second one. Lots of genes do this chromatin remodeling. There are actually lots of different ways to remodel chromatin. Our favorite is going to be this histone acetylation, but we're going to learn a little bit of other examples of chromatin getting remodeled. One thing that we've already talked about a little bit in our course is DNA methylation, and that represses gene expression. And there are 4 letters in the DNA alphabet, and cytosine can get methylated, can have a methyl group covalently bound to it, and that represses gene expression. And that's considered an example of chromatin remodeling, partly because chromatin remodeling is usually facultative. That means it could change, and not constitutive. That means it's not changing, it's heterochromatin. And so things that are changeable, like the methylation of DNA, are changing. We have to be careful because our authors do say, and it's true, that in addition to histone acetylation, one thing that can happen is they can be methylated. So a methyl group could be added to histones, and that can sometimes increase or decrease gene expression, so it gets complicated really fast. And like I said, there are several ways to modify histones, and we're going to focus on acetylation. That's plenty. When the histones get acetylated, they are going to open the chromatin. Euchromatin allows gene expression. When they're going to deacetylate it, closed chromatin, heterochromatin represses gene expression. Then the third example we're going to talk about is going to be in that thing when we talk about that other new transcription factor, heat shock transcription factor. And in that example, we'll find a third example. And those are the 3 examples of remodeling chromatin we want to be ready to write about on an exam. So euchromatin is permissible for transcription, and heterochromatin is more compact and blocks transcription. Okay, so, and again, this is chapter 7 we're talking about. So here, we're not representing any of that chromatin remodeling, because in our example, right, back to the clay, really literally what the MyoD does is it interacts with tau-binding protein, TBP. And we didn't label it or talk about it because we talked about plenty in the original DNA clay model videos. But it doesn't have to unfold the chromatin. The chromatin has histones bound to it that are already acetylated, and we just don't represent them there because it wasn't a factor. So, different story. Okay? So, then here's that figure again showing that, and it's showing the enzymes. So, when, in our case, the estrogen hormone is present, this is the enzyme that's bound to the estrogen receptor. And this is histone acetylase. The full name is histone acetyl transferase. I usually just call it histone acetylase. Then here, in the absence of the hormone, the HAT, the histone acetylase, is let go, and instead a histone deacetylase is bound. You see it's tethered and removed all these acetyl groups enzymatically over several minutes. Transcription is repressed, the chromatin is more folded up. And these two, they're transcription factors. Yeah? But they don't bind to DNA. Remember, we just learned that estrogen receptor has a DNA binding domain. These 2 enzymes do not have a DNA binding domain. They bind directly to the estrogen receptor. And so there are some transcription factors that bind to DNA and have a DNA binding domain. Then there are these transcription factors and many others like them that don't bind directly to DNA. They are transcription factors because they're regulating transcription, so they are transcription factors. The enzyme, and these are enzymes, and not all of the proteins that are transcription factors that don't bind DNA are enzymes, but these are, in general, transcription factors that don't bind DNA directly are called co-regulators. Because they have to work with a helper that's bound to DNA. And what's happening is that the DNA binding enzyme, transcription factor, that's bound to the DNA, so that's helping tether the co-regulator. So it's kind of like the transcription factor is kind of like the collar that is attached to the leash of the dog, and this enzyme, the co-regulator, is the bitey dog in this case, right? Does that make sense, right? We have the DNA, and it's like a leash tethering this bitey dog. Well, the estrogen receptor is really just a collar to collar that dog to the leash. And it's the dog, it's the co-regulator that's the scary transcription factor activity. It's the thing that's going to change the rate limiting step. Because remember, we're still focused on that. This is changing the rate limiting step, and it's that we need to change from heterochromatin and deacetylated histones that are packed away to acetylated histones, open chromatin. And so that's what's happening here. And you can see how this is an awesome transcription factor because it's repressing transcription factor and then it's activating it. So it's repressing for years and years, right? A child's born and goes through childhood and then reaches puberty. And during that time in childhood, the estrogen receptor target genes are all packed away. So it's a very good example, very memorable example for us to learn about. So these co-regulators, they always are either an activator or a repressor, and that's what we have here. And so people rarely use the term co-regulator because they instead say, well, this is a co-activator and this is a co-repressor. So we got to learn those terms. And we'll learn the term co-regulator because that's the general term. Okay, so I think we're already almost set with this. A little bit more. So here's just showing the acetyl group being added. So it's real, chemistry, and a covalent bond is formed there with that lysine like that. And we have the enzyme histone acetylase adding it, and the enzyme histone deacetylase removing it. And these are part of the histone tails. And what's happening really effectively is that this is charged and causes the protein to fold up more than this when it loses that charge. So it just has to do with the structure of the histone proteins, and it's at the end of the histone protein. Here's the histone protein stretched out, and people refer to this as a tail, and you can see there are multiple lysines along here, multiple charge groups that either are acetylated or not, and when they're acetylated, they tend to unfold the chromatin, and when they're deacetylated, they fold up the chromatin. So a lot is known about the molecular details of that. Okay, so histone acetylation generally increases gene expression. Okay, so then I've talked in class about how a person can get injections of estrogen or testosterone, and I mentioned that in males, estrogen receptor is working to repress the expression of target genes. If a male gets injections, and not just a human male, but any vertebrate gets injected with estrogen or testosterone, they can start to develop different secondary sex characteristics. So we're going to talk about a drug that's used to treat cancer, and it's called tamoxifen, and it has lots of different trade names. And this is going to be something that comes up in chapter 19, the last chapter in our book, that we'll get to pretty quickly, although not before the first exam. So this is a drug, and it's an antagonist of estrogen. So we have to learn 3 words here. So an antagonist inhibits a receptor. An agonist, it's a snow day. An agonist activates a receptor. So we got to learn those terms. And so an antagonist and an agonist, they have an additional specific word and or feature, and that is they do that by binding directly to the receptor. There's another way to inhibit the receptor, like the estrogen receptor, and that would be to somehow inhibit its expression, so we don't even make the protein. And that wouldn't be an antagonist, that would just be an inhibitor, but we wouldn't call it an antagonist. So antagonists and agonists come with very specific terms, and that's important because tamoxifen is an antagonist, and it blocks estrogen by not inhibiting expression of the estrogen receptor. The protein is there. It binds directly to the protein, and it inhibits its activity. And so by inhibiting its activity, the protein is going to be more folded up. Because tamoxifen works by inhibiting the histone acetylase binding, so it can't open up, but it doesn't block the deacetylase, so the protein is still inhibiting genes. So it's an inhibitor of the estrogen receptor, and it's used to treat breast cancer. And I don't think tamoxifen is considered a cure, but it works really well and helps doctors have more time. And here's an example of that. And this will be, I see we're going to have run out of time today, but here's an example of that. When we get to chapter 19, we'll have some examples of, cancer patients. And so I know someone who had breast cancer, and they had surgery to remove the tumor. And the doctors tried just to take a small amount of tissue away, and they do that. They take away the tumor, and they also remove a little bit of tissue that was remaining after the surgery, and they call that the margins, the edges. So these are the edges that were beyond the tumor that should be healthy cells. So they did that surgery, and then they test the margins. And when they tested the margins after this person's breast cancer surgery, the margins were said to be not clear. That's how they say it. The margins were not clear. That meant they found some scattered cancer cells. They hadn't removed the whole tumor. So they did a second surgery, removed some of that, more of that tissue. Again, trying to remove a minimal amount of tissue so the person would be healthy and go on. And after the second surgery, the margins were still not clear. So they did a third surgery, and now the margins were clear. And those surgeries were done years ago. The person seems to have been cured of breast cancer. Okay, now they want to do those surgeries carefully and remove a minimal amount of tissue. So it took 3 surgeries. The reason they were able to take their time and do the 3 surgeries is because that person was taking tamoxifen. So again, tamoxifen isn't really considered a cure, but it will put the cancer into remission for a long time and give the doctors a chance to treat the patient carefully and make sure they treat the patient well. Okay, so tamoxifen is antagonist. It binds directly to the receptor. So when any person ever has breast cancer, they check to see if the person is expressing the estrogen receptor protein. That's ER. And if they are, they say it's an ER positive cancer. So all cancers are either ER positive and about 70% are, or they're ER negative, about 30%. And they use tamoxifen to treat ER positive breast cancer. They don't use it for this because these people aren't expressing the estrogen receptor. In ER positive breast cancer, the cancer is almost always growing in an estrogen dependent way. And the estrogen receptor is activating genes, and some of the genes are stimulating the growth of the breast tissue and the uncontrolled growth of the breast tissue. So that's a really important concept in molecular biology. It's all matter-of-fact cause and effect relationships. And I mentioned that there was a third term. So if we say antagonist or agonist, we know specifically that this is working by binding to the target protein. The third term it's called a mimic. And a mimic is where it's doing the same job as the hormone, but we're not sure if it's doing that by binding the receptor or through some other pathway. And if it's inhibiting, and we don't know for sure if it's an antagonist, we usually just call it an inhibitor. Oh, and that's my little timer saying that we have like 5 minutes left in our official class time. So we'll talk more about tamoxifen because it's really our first cancer drug, and we'll only learn about a few, but it helps us understand the receptor activities like that. So tamoxifen is bound to the ligand binding domain of the estrogen receptor directly. It's directly inhibiting the estrogen receptor activity. It's an antagonist. And with that slide, we'll stop here today. All right, great. So you need to take notes on all of this and learn this as well as a regular class, and maybe even better because it's a snow day. So you hopefully have some time to play in the snow that isn't even falling yet, but will fall a bunch this afternoon. And then also remember we have a homework assignment. Great. All right, that's it for now. Thank you.